Hello everyone, we are back for our last session and our last academia talk of our conference. So with this, actually, we have the pleasure to close them with Karissa, Dr. Karissa Sanbonmatsu. She earned her PhD in astrophysical sciences at the University of Colorado, Boulder, before moving to Los Alamos National Laboratory as a postdoc scholar. Now, Karissa is a group leader at the Los Alamos National Laboratory, and her team applies a variety of computational and experimental approaches to understand the mechanism of a diverse array of non-coding RNA systems, including ribosomes, riboswitches, and also long non-coding RNAs. She and her group were the first to elucidate the secondary structure of an intact long non-coding RNA and perform a biomolecular simulation of an entire gene. Karissa, our virtual stage is now yours, so you can basically share your screen. And let's hope we can see it. Yeah, thanks.
Okay, can you see the screen?
I think it's loading. Okay, now I can see it well. Cool.
Okay, great.
So we are looking forward to your talk.
Well, it's an honor to be here speaking. Just turning the stopwatch on. Sorry. Okay, it's an honor to be here speaking at the Horizons Conference. I'd like to thank the organizers for giving us the opportunity to share our work. And I'm going to talk about this new class of RNAs called long non-coding RNAs and the role of these RNAs that they play in epigenetic phenomena. So more specifically, I'm going to talk about 2D and 3D structure-function relationships for individual long non-coding RNAs. Well, the main application area for these RNAs is epigenetics. And what is epigenetics? Well, all cells in your body have nearly identical DNA sequences. But the question is that how can the cells be so completely different if they have almost identical DNA sequences? So, for example, muscle cells, blood cells, neurons, pancreas cells, and so forth have vastly different structures and ways of operating, but they have nearly identical DNA sequences. Well, the answer is something we call epigenetic programming. And so in particular, what happens is that the genes are turned on and off differently as the embryo develops, and they're sort of synchronized and turn off and off in just the right times and just the right places to enable vastly different cellular structures and different processes. And so epigenetics is the science of how this switching process works. And more interestingly, these epigenetic effects also play important roles in disease, in brain function, and they can also actually be persistent over many generations. So here I'm showing just a little tiny piece of a chromosome. This is an atomistic model we made. And the idea is that compacted regions of chromosomes tend to silence genes, whereas more expanded regions of chromosomes have the tendency to allow the genes to be expressed. So the idea is that if it's highly compact, there's not much room for the apparatus, the different protein complexes and so forth to get in and start the process of gene expression. And it has a tendency to dampen the gene expression. So in terms of epigenetics, there are many different open questions. One of these questions is the mechanism of targeting and recruiting epigenetic factors to specific sites on the chromatin. And so here the idea is that these different complexes of proteins bind to the chromatin, which is the DNA wrapped around these little molecular spools, but they have to bind in the right place at the right time. And people don't really understand how that can be. How can this piece of chromatin sense that stress hormones flowing through the cell, or sense that the embryo is at a certain stage of development? This is really a big mystery right now. And so the main idea is to try to figure out, what triggers the recruiting of these factors. Some other open issues are demethylation. So it turns out in epigenetics, there are different what we call marks on the chromatin that trigger this collapse. And so these can be methylation of these molecular spools called histone proteins, or methylation of the DNA itself. And the unmethylation or the demethylation is really an open question in the case of DNA. There's been a lot of progress made, but it's still an active area of research. And then also the mechanism of stem cell reprogramming is not fully understood. And then another big open area is the inheritance of these modifications from one generation of cells to the next generation, and also one generation of the whole organism to when it has offspring. The signals, the marks can be transmitted, but people don't quite understand how this works, especially in the case of the histone proteins. And then finally, these are all processes that are important therapeutic targets. So a lot of the drug companies now are racing to find better therapeutics to control these kinds of processes. And in the case of RNA molecules, one of the main focuses is on this mechanism of targeting and recruiting. And so it turns out that this emerging class of RNAs called long non-coding RNAs is thought to play a role in this recruiting process. So it's known that these RNAs can sense small molecules for example, in the case of riboswitch RNAs. And the idea is maybe they could sense some of the environmental cues going on in the cell, fold a certain way, and recruit complexes. So a lot of people are focusing on long non-coding RNAs for recruiting complexes to chromatin and other mechanisms such as sponging out epigenetic factors and various different mechanisms. The abbreviation for long non-coding RNAs is the LNC RNAs, and there's also long intergenic non-coding RNAs, and both of these we tend to call lincRNAs. The long non-coding RNA is defined to be any non-coding RNA above 200 nucleotides, and it's typically referred to if they're involved in epigenetics in mammalian systems. Then you typically call it a lincRNA, and then they can be as big as hundreds of kb, and they're typically range from say 1 kb up to 10 kb as a typical lincRNA system. They often look like coding RNAs in the sense that they're polyadenylated, they're capped, they're spliced. They look like a sort of a normal messenger RNA, but they don't they in sort of over 90% of the cases, they don't make any protein, and they have an RNA function, and they can act in trans or in cis. And then they're often associated with epigenetic effects, such as these methylation marks on the chromatin, like histone 3, lysine 27, trimethylation, and different epigenetic marks. So many of these lincRNAs have been shown to have important visible phenotypes, the most famous of which is EXIST, and this RNA plays a very important role in the development of females. And so for females, we have 2 X chromosomes, and one of the X chromosomes is actually totally silenced. And it turns out that it has to be totally silenced for proper functioning of the cell. And if both X chromosomes are turned on, then the organism isn't viable and the embryo dies. So it's very important that this X chromosome is almost totally silenced. And the way the chromosome gets silenced is that these long RNAs are manufactured, and they sort of physically coat the chromosome, and they trigger this process of inactivation of almost all the genes on the chromosome. And so the RNA that does this is called EXIST, the X inactivation stimulated transcript. Another example of a lincRNA is Braveheart. This was an RNA discovered by Laurie Boyer's group at MIT, and this plays a key role in the development of the heart. Another one is Cool Air. This is a plant RNA that senses the cold, and it's important for flowering in the springtime. This was discovered by Carolyn Steen's group. Another one is Link BRN1B. This is important for neocortex development. And then another one is called Megamind, that's important for vertebrate development. And there are many others that have been found that have very important phenotypes. So it turns out that up till now, most of the field of long non-coding RNAs has proceeded in the way of genome-wide studies. So they're looking at genome-wide. They want to see the global picture of when large numbers of long non-coding RNAs are being expressed, under what conditions, if you add estrogen or different things to the cell, what happens to the global picture of like thousands of these long non-coding RNAs as a whole. But there are very few groups that have been looking at these one at a time. And on the screen, I'm showing a Japanese proverb that states a difficult task cannot be done quickly. It can only be achieved gradually by small steps, a little at a time. And this is at one of the more prominent temples in Japan, and I think it has about 1,000 steps, and then it's written on the side. So the idea is that as you walk up these 1,000 steps, you're supposed to learn this lesson that the difficult tasks need to be accomplished one step at a time. And that's how we're proceeding, is we're trying to look at each long non-coded RNA one at a time and try to understand its mechanism as much as we can. And then after we study a lot of these, maybe we can figure out some general principles of these long non-coding RNAs. So here I'm showing an atomistic structure of the human ribosome using cryo-EM. And this you could consider sort of the original gangster of long non-coding RNAs, in that it's about 5 kb in length. It's non-coding, so it's a non-coding RNA, a long non-coding RNA, and it makes a long non-coding ribonucleoprotein complex. And so what we're trying to do is to apply techniques from the ribosome field to this new emerging field of long non-coding RNAs. And so my group is mostly a ribosome group. So we've been working on ribosomes since about 2001. And we know the history of the ribosome field very well. And the ribosome is one of the probably better studied RNA complexes in the cell. And so in the RNA field, first they came up with the sequence early on, and then in the 80s, they got the secondary structure. And so the way they did this was using a variety of chemical probing techniques combined with phylogenetic sequence analysis. And then from that, Harry Noller, Carl Voice, and Robin Guttel came up with this 16S ribosomal RNA secondary structure. And this is one of the cornerstones of phylogenetic analysis today. And this is showing a nice example of a complicated secondary structure for a long non-coding RNA. And it basically gives the pattern of base pairs in the RNA. It's a 2D map giving the pattern of helices in that RNA. And I'll go into that in more detail in a few minutes. And then in the ribosome field, after the secondary structure was solved, then people started to go for the 3D structure. And the first step was to do scattering. Or just to back up, so my own group that I'll be talking about in this talk, we've used these secondary structure techniques to look at a variety of different long non-coding RNAs. Okay, so back to the history here. So the next step in ribosome studies was scattering. And so here, people such as Peter Moore's group used neutron scattering. And what they were able to do was to try to figure out in 3 dimensions where the different proteins were positioned. In the bacterial ribosome, there's more than 50 proteins that get intermixed into this RNA to sort of glue the RNA together. And through scattering studies, they were able to get some kind of very coarse picture of the morphology of the ribosome and where the protein's bound. And then after that, over the course of about 3 decades, people used X-ray crystallography and cryo-EM to come up with high-resolution structures of the ribosome. And then once you have the structures, you're only beginning to understand the ribosome. You're not done at all by any means of the investigation. So once you have the structures, you need to figure out how it works, because when you have the structure, you don't know how it works at all. You just know what one snapshot in its life cycle looks like, or you could have many snapshots, but you still have no clue about the mechanism. So the next step is to figure out the dynamics, and you can use single molecule FRET, and you can also do large scale molecular dynamic simulations to see what the mechanism is. You can look at the entropy and the enthalpy, exactly what's happening to cause the different conformational changes to make the ribosome do what it does. So there's sort of a long list of things we need to accomplish for long non-coding RNAs to get to the level of understanding where we are for ribosomes. And so right now, there are many sequences available for long non-coding RNAs. We've determined a few structures, and some other groups have determined a few structures. And now we're moving into the scattering stage of investigating the structure and function of these long non-coding RNAs. So in terms of the fundamental questions, for these long non-coding RNAs, there are many open questions. Some of the ones we're focusing on are, do the long RNAs have a well-defined 2D structure or secondary structure? Are the long non-coding RNAs conserved in terms of their structure? Are there structure-function relationships we can figure out? And then what do these individual lincRNA systems look like in 3 dimensions? And so the strategy we're taking is we're following the steps of what happened in the ribosome field, and then we're trying to do a modern twist by borrowing techniques from the riboswitch RNA field. Okay. So the first question is, do lincRNAs have well-defined structures? So there are different possibilities you can think of when you have a huge RNA flying around in the cell. It could be fully disordered, it could be highly compact, or it could be somewhere in between. The fully disordered situation we can kind of rule out because people in the RNA biochemistry field know that even if you don't have Watson-Crick pairs, you can have lots of non-Watson Crick pairs happening, and so basically we'll probably get some kind of secondary structure no matter what. It's going to curl up into some kind of ball or some kind of situation where you have lots of base pairs all over the place. So basically the question is it highly compact, or is it kind of a mixture of compact regions and extended regions? And so, as I mentioned before, there are many groups out there doing genome-wide studies, and then a few of us are trying to look at these one by one. And then here I'm just showing a sort of artist's impression of what these 3 different scenarios might look like. So it turns out that if we, in the long term, if we wanted to go for 3-dimensional structures, we need really purified samples. So in particular, if we want to go to high resolution. So in the case of, for example, crystallography, you often need a reconstituted system if you're going for solving one of the first structures of these. So it makes sense to go look at if you're going for high resolution structures, to start with in vitro systems before trying to figure out what they look like in living cells. I'll just flip through these quick because I'm going a little slow here. So the next question is, are the long non-coding RNAs conserved? So if you have a typical protein amino acid sequence, you often think of a highly conserved protein as maybe around 30% sequence identity. Now, for nucleic acid sequences, if you just look for something in BLAST, the defaults are set to 95% sequence identity for 40 nucleotide stretch. And some RNAs do have this high level of sequence identity, such as the microRNAs, which regulate gene expression, or the ribosomal RNAs. However, there are a lot of RNAs that have much lower sequence conservation, but they have high structure conservation. So even though they have almost no sequence similarity, they're still conserved across thousands of species and with similar functions and similar structure. So examples of these are the riboswitches. These have 50 to 65% sequence identity. So most of these you would not find if you do a BLAST search. But they're conserved across thousands of sequences. Spliceosomal RNAs U2 and U4 have sequence identities of less than 60%, but they're conserved over 9,000 sequences. Even the 5S ribosomal RNA, 60%, but conserved over 200,000 sequences. So a lot of these RNAs, you won't find them in BLAST because they don't have much sequence identity, but they have highly conserved secondary structure, 3D structure, and function. So one of the misnomers is that these lincRNAs aren't conserved, but I would maintain that you don't know if they're conserved or not by just running BLAST. You have to do a lot of detailed biochemical analysis to find the 2D structure. That's step 1. And then after that, then you can begin to ask the question, is it conserved or not conserved? Okay, so just to give you a quick rundown of RNA secondary structure, these RNAs are organized in terms of double-stranded Watson-Crick helices typically, but it's not like DNA in the fact that you can have all kinds of funky, almost like broccoli or tree-like branching structures happening. So in this case, we're looking at the S-adenosyl methionine riboswitch. This regulates genes in bacteria by detecting that ligand as adenosine methionine, and it's organized in 4 helices. And so on the left-hand side, we're showing the base pairing structure with the 4 Watson-Crick helices, and then connecting the 4 helices is what we call 4-way junctions. So they're little looping regions that kind of fold up and crinkle up, and they often form like intricate tertiary structures with lots of hydrogen bonds and magnesiums mediating them and so forth. And you get this complicated 3D structure on the right. This was solved by Rob Beatty's group in Colorado, and to give you this active state of the ribose switch. In the middle is how it binds these ligands. And so here's another example, the TPP ribose switch. In this case, it has a sequence identity of 57%, but it's conserved over 11,000 sequences. So it has quite low sequence conservation. And so here on the left-hand side in red are the conserved sequences, which is not much compared to the full size of the RNA. But on the right, the secondary structure is highly conserved. So the red are the conserved helices and therefore conserved loops in between them. Okay, so to study these secondary structures, we do this technique called chemical probing that they did in the ribosome field back in the day, and then also in the ribose switch field. And the most the simplest technique is inline probing that Ron Breaker used a lot to come up with secondary structures of ribose switches. And here you just make your RNA in a test tube, you let it sit overnight, and then it incubates, and then the highly mobile regions tend to undergo cleavage, and then you can map out which nucleotides are cleaved and which are not cleaved, and that gives you a map of the base pairs of that RNA, so which bases are paired and not paired. And then Kevin Weeks came along in 2005 and came out with the selective 2' hydroxyl acylation analyzed by primer extension. This is shape probing. And here you add a reagent. On this diagram, we're showing NMIA. And that reagent has the special property of forming covalent bonds only to highly wiggling backbones. So basically it detects the residues in the RNA that are highly mobile. Their backbones are highly mobile. And that gives you another readout of the base pairing pattern. And then the more classical techniques used for ribosomes are DMS probing that detect the face of the base. And also hydroxy radical probing can give you some information about protection of tertiary contacts. And so how does this work in detail? So the idea is that if you have one RNA sequence, it might have many different possible folds. It won't just have one fold. And so, for example, in this case, I've drawn an example where one sequence has 2 possible folds, one long helix and another possible fold has 2 smaller helices. And then if we go in the lab and make the RNA and do the chemical probing, we can get this reactivity readout, which gives us an idea where the looping residues are. And then this can lift the degeneracy and allow us to narrow it down to this structure with 2 helices. And then finally, what we can do then is do phylogenetic analysis against other species and look for what we call covariant base pairs. So these are regions that have double simultaneous mutants that preserve the Watson-Crick base pair. So the CG might doubly mutate to a GC, and that gives us a little more confidence in this secondary structure that it's conserved over species. Okay, so next we can talk about the validation of these secondary structures. So in the ribose switch field, these secondary structures were determined just with test tube biochemistry. So just synthesizing the RNA in the lab, doing the chemical probing assay, coming up with the 2D structure. But then it turns out that all of these ribose switch secondary structures were validated with high-resolution X-ray crystallography by Rob Beatty's group in Boulder and Dinshaw Patel in New York. And so for these ribose switches and many others, high-resolution X-ray structures were solved. And nowadays, cryo-EM structures are being solved for these ribose switches. And they've all confirmed this test tube method of determining RNA structures. And then the next step is to look at the dynamics of these RNAs. So you can look at different issues like induced fit versus conformational selection and binding to its ligand partner. And you can also look at, it turns out that a lot of these ribose switches, if they don't have the ligand, they're quite floppy and flexible. So you can get a feeling of what the envelope describing the flexing of the helices looks like in 3D using small angle X-ray scattering to get 3D envelopes of these. Okay, so all these techniques are great for small RNAs, but how about for long RNAs? Well, for long RNAs, we can again do the chemical probing, but the problem is we know which bases are, we know which bases have low reactivity, and therefore they probably have a base pairing partner, but we don't really know what that base pairing partner is. And so for a long RNA, you can have hundreds or thousands of folds that are consistent with that chemical probing signal. But the question is, how do you eliminate the sort of false positives and isolate one particular fold? And so you can think of this as analogous to if you're having a wedding and you're planning the wedding reception, you have a lot of people coming to the event and everyone sits at different tables, but you may have enemy families coming to this event that may end up arguing or getting in fights and so forth. So you want to organize the seating plan so that everyone gets along and you don't have any, you want to minimize the drama at the event. So here you'll have to work out a precise seating plan so that everyone gets along. And if it goes to large numbers, it can get quite exasperating. And there can also be confounding factors. Like I went to one wedding where my brother came and he got there early and he didn't want to sit next to one of our cousins. So he swapped the name tags on the table. So therefore he didn't have to sit with this person he didn't want to sit next to. So there can be other confounding factors that make things even more complicated. Okay, so the way we try to solve this problem is we use a technique called shotgun secondary structure. And this is sort of a divide and conquer style. So what we do is we do a chemical probing experiment of the full long non-coding RNA. That's step one. And then we do successive rounds of chemical probing on smaller fragments of the RNA. And then what happens if we can find a signal for the fragment that is a close match to the signal of that same stretch of RNA in the context of the full RNA, this is really good evidence that that piece of the RNA has an independent modular fold that stands on its own, even in the context of the full RNA. And then if we can find multiple situations like that, we can come up with subdomains that we know fold independently, even in the context of this huge RNA, and then piece them together to put together the fold of the full RNA. So what we do is we synthesize the RNA with just using runoff transcription. Then we fold the RNA, we do our structure probing, and then we do reverse transcription, and then we visualize with capillary electrophoresis. And this is a pretty standard technique that a lot of people use nowadays. And this has been ported by our group and others to working on Illumina high throughput sequencing systems as well. So the first example I'll show you is called cool air. So in the case of cool air, this is an RNA discovered by Carolyn Dean's group that has the function of sensing cold. So it's sort of a repressor of a repressor. And it turns out that in the fall, this gene called FLC turns off flowering when it's getting cold out. But then in the springtime, an RNA represses that repressor and allows flowering to happen. And it turns out that this RNA is only active after 4 to 6 weeks of cold exposure. And Carolyn Dean, who discovered this molecule, came to us and asked us to work out the secondary structure of this system. So what we did is we did shotgun secondary structure probing. And in blue, we're showing the probing signal for the whole RNA, and then red, 3 different fragments. And so we find that in the case of fragment number 1, the first half of that in pink, we saw a nice match between the probing signal of the fragment versus the probing signal of the full RNA. And so therefore, we conclude that that probably forms a modular fold, even in the context of the full RNA. And that's shown here on the right-hand side, the secondary structure, where just regular letters represent low reactivity, but the colored orange letters represent high reactivity in the signal and are on the looping regions. And then the blue region is this mismatched region, which probably does not form a modular RNA and probably forms pairs with long distant pairs with other parts of the RNA. And then for the second fragment, we saw, in this case, it matched almost over the whole fragment. So we came up with this corresponding secondary structure. And then when we put everything together, we get this fold for the full RNA. So here we did some analysis and we saw some interesting shapes here, or secondary structure motifs. This one has a very large looping region on the 5' side. And we saw that here again, and we're calling this a right-hand turn motif, or an R-turn. And then in addition, we see a multi-way junction in the middle and also a 3-way junction toward the left at the 5' end. And so we wanted, before we came along, cool air was thought only to exist in Arabidopsis thaliana, but we wanted to see if we could find this in other species. And we went ahead and did shape probing on Brassica rapa, and we also saw a very similar secondary structure with the 2 right-hand turns and the multi-way junction. And then a student in Carolyn's lab did RT-PCR for several species of Brassica, and she could pull out this RNA. And so we found this in 5 other species, and we came up with this consensus secondary structure showing only base pairs that are conserved across all the different species. Okay, so I think I'm running out of time, so I'm going to skip over this RNA. This is the steroid receptor RNA activator. It's important for the estrogen response, but I'll just flip through this really quickly, where we did the 3S shape probing on this, and we found 4 subdomains. We did 4 different methods of probing, and they all weren't consistent with each other. We saw a lot of covariance. Other people looked at binding to different parts of our structure. This is an example of our CAP-UP. And then we found a right-hand turn motif in this secondary structure as well. And this was, the domain organization was conserved across lots of different species. And this is showing how the loops are conserved and how it's modular. Okay, so then the last system is the Braveheart system, and this was discovered by Laurie Boyer's group at MIT. And this is around 540 nucleotides, and it's important for cardiovascular lineage commitment in mouse development. And so we did our 3S method on this, and we got this nice secondary structure where, again, we see the right-hand turn motif. We saw a multi-way junction and 2 other helical regions. Here's the right-hand turn motif. And then Laurie Boyer at the time, CRISPR just came out down the hall from her lab, and she wanted to try it on this Braveheart. And she was asking which part should she do CRISPR on. And so we told her to look at this right-hand turn motif. And so she deleted just the looping region of the right-hand turn in her mouse studies. And we also deleted it in our chemical probing studies. And when we deleted it, interestingly, we see the exact same secondary structure, just minus that loop. And it doesn't have to come out like that, but that's what we got. And then she deleted the right-hand turn in her mouse studies, and she found that deleting the right-hand turn had the same effect as knocking down the entire RNA. And so she has this nice beating embryoid body assay, and she saw when you delete just the RNA turn, the R turn, that it knocks down that phenotype. And then she did a lot of epigenetic assays to figure out what stage of development this was happening in. And she also figured out that it had a protein binding partner, a zinc finger transcription factor called CNBP. Okay, so just going to the final part of the talk, the next thing we did is we did 3D studies of this RNA, Braveheart, and also Braveheart complexed with CNBP, the protein. And this was a collaboration with Chushar Patel's group in Canada, and also Ivo Hoffacher's group in Vienna, and Dunam Kim did the work on this. And so here we use small angle X-ray scattering, where you have an X-ray source. You then collect you scatter it off the sample, you collect the data and reduce it to a 1D profile using radial averaging. From that, you can get a probability distribution. And then from that, you can get a 3D volumetric model. And so we used the B21 beamline at Diamond in the UK. We collected the SAX data. We used scatter and ATSAS to process, and we had a size exclusion chromatography SAX set up to help obtain a model dispersed sample. And then in terms of the 3D volumetric reconstruction, we used the Damon software package, 12 models per sample, and we used Demiver to rotate and align the models in the end. And so we developed this complicated pipeline where we had on the left a pipeline to get the 3D volume model from the SAX data. We had a pipeline to get an atomistic model using our 3S shape combined with a coarse grain modeling technique and then coming up with sort of the scaffold of the 3D structure, and then threading that with 3D secondary structure motifs in the protein data bank, and then stitching them together. And then we also had a null hypothesis structure where we shuffled secondary structure elements just so we could have an additional control. And then we did chi-squared with chrysol to check things. And so in the end, what we see is when we look at the scattering curves that when you add in the protein, you get a compaction of the overall structure. And also when you add magnesium, you get a slight compaction. And that's typical of these RNA systems. And then here I'm showing a diagram of the goodness of our 3D model. And so on the left-hand side, left-hand axis is a chi-squared, and on the right, on the x-axis is a scoring function. And so basically we had thousands of models. In the upper right corner are the bad models where we didn't get good fits. In the lower left are our best models shown here. We got a nice correspondence between the model and the experimental data. And so in the end, we took 10 of our best models or so, and we could see they have a lot of flexibility in them, but they all have the same secondary structure and similar 3D motifs. And then when we superimpose those models and take a probability distribution of those, we get a quite close agreement with the 3D volumetric models from the scattering experiments. And then we also looked at the volumetric models with and without the protein, and we do think there's some kind of reorganization happening when you add in the CNBP protein. Okay, so that's it. So to conclude, we have this sort of divide and conquer technique where we probe the full RNA and then we probe fragments. We look for similarities and we can pull out all the subdomains for 2D structures of lincRNAs. In cool air, we could see that one of these secondary structures was conserved across species. Then in CRISPR studies, this R-turn that we pulled out was shown to be essential for the function of the RNA. And then in 3D scattering studies, we not only could validate the 2D secondary structure, but we could get a picture of, to begin to see what the 3D structure of the RNA looks like and that it changes upon the protein binding. So I'd just like to thank everyone who did the work. So Irina Novikova and Scott Henley did most of the early studies of the lincRNAs. Dunam did the 3D study and the SACS chemical probing and the modeling. The collaborators, Chushar, Ivo, Lori, and Carolyn, are funding sources. And then lastly, we're recruiting a number of postdocs right now, so please contact me if you have any questions. And thank you for your attention.
Thanks for the great talk, Karissa. We have many questions coming up. So the most voted one was from Margarita. She's asking, what are the current views on the role of riboswitches in evolution? Could they indeed have emerged as pre-protein enzymes?
Pre-protein enzymes, yeah, that's a great question. I think this is a possibility. There's not a lot of studies that have been focusing on the evolution of these riboswitch RNAs and how they evolved, but I think a lot of people think that if there was an kind of RNA world style system in the early days, these could have been the precursors to metabolism, possibly.
Great, thanks. The second question is from me. So I'm wondering, what are the limitations on the sample preparation of long non-coding RNAs for cryo-EM?
That's a great point. So even for the shape probing experiments, it's tough to make RNAs past 5 kb. I would say that's really pushing. You have to be a really good RNA biochemist to pull off that, I'd say. So going to like 100 kb is really not hard to conceive right now in test tube experiments. And so for cryo-EM, there's upper and lower bounds. So Riju Das's group published structures of riboswitch RNAs recently at low resolution, which are 100 or 200 nucleotides. And then I think you can make an RNA of a couple kb in the lab. So I'd say that would be a good range right now to stick to. And you could try going with lysate as well, but I think this is much tougher than as opposed to making the RNAs in the lab.
Great. Alvaro also has a question, and he says, great talk so far. How does their flexibility relate to difficulties in their crystallization or cryo-EM analysis, which I guess it's actually related to my question.
Yeah, this is a great question. So again, in the Riju Das paper, they comment a lot on this on the cryo-YAM. So interesting in that paper, like the typical notion in the riboswitch field is that if you don't have the ligand, it's very floppy, and we can see this on the gel. When you run a gel, it really has a massive shift when you add in the ligand. And in Sachs, it shows to be floppy. But they were finding it. Some of them without the ligand were higher resolution actually. So it's I feel like you never know what you're going to get with these RNAs and the flexibility of them. But that's certainly an important issue.
Also, Barbara is asking, in the structures you have shown, there were a lot of right-hand turn motifs. Do all right-hand turn motifs have a similar function, say like homologous domains of proteins?
Yeah, nobody knows right now. So the only one that's really been studied is this one of the Braveheart, and we believe the Laurie's CRISPR analysis. She has some evidence that it binds to the protein, and then we showed in Sachs that that the RNA binds to the protein. So we think it could be responsible for the protein binding, but in the other RNAs, these right-hand turns haven't been studied. There is a similar motif in spliceosomal RNA where it binds proteins in the splicing RNA system. But it's a very rare motif, so we've seen it mostly only in the long RNA systems.
Cool. So as a last question, Florian, he's saying thanks for the interesting talk, and also he's asking, focusing on the distribution of epigenetic effects in whole tissues, are there mechanisms to distribute them throughout organisms? And then there is, well, he's saying, or does the long-term phenotypic homogeneity or epigenetics originate from mitosis-based distribution of single cells rather than from intercellular signaling?
Yeah, that's kind of over my head. I'm not really a cell biologist, but yeah, I can't really speak on that too much. I mean, somehow these effects propagate, through the tissue, and I mean, this is not. I'm not really an expert in this, but I can just mention to the other viewers, not to the person asking the question. A more basic thing is this Waddington's landscape, where the idea is that during development, different decisions are made, and you can think of it as like if you're skiing down a ski slope, and as you go down, every once in a while you have the signs, and you have to take the trail to the left or to the right. So this decision making happens in development, and I think a lot of these decisions get frozen in during those stages of development, so that the whole tissue will have a certain epigenetic switch thrown possibly, and then it specializes during development. But yeah, I have no idea about the answer to this question that was asked though.
All right. Okay, so thanks once more for the great talk and for being here today, Karissa. I would like to make a small announcement for our participants. We are going to have the closing ceremony of our conference in a few minutes. And we wish that you're all there in a few minutes with us. And we will basically also be announcing the awards for our posters awardees. And we hope to see you there. And thanks, Karissa, once again for joining us.
Thank you so much. I really enjoyed it. Thank you.
Bye.
Bye.